Hello, in our second lab, we're going to talk about macromolecules, and we're going to focus on three main experiments. In the first experiment, we will study a test called the Benedict test that study reducing sugars. And we will use different test tubes and we will use hot plates and we will warm up the sugars here. And if they are negative, they will be blue. If they are not reducing sugars, and if they are red, they will be positive for reducing sugars, and in between we have intermediate degree. In our second experiment, we are going to learn how to do a Bradford assay, that is an assay to estimate the amount of protein. And again, we will have, if it's brown, in this case it will be our negative result, and if it's positive, it will be blue. And the higher the concentration will be, the more blue the test tube will look like. And then finally at the end, we will learn how to make a standard curve for the Bradford assay, and we will use a spectrophotometer to do that. So let's start today. First thing that I have to tell you about the Benedict test is that you have to be careful with two things. First, be careful with the hot plate. Yeah, I don't think that is very dangerous. It's like cooking, so it's going to be fine. Just be very careful with your hand. It's going to reach almost 100 degrees, so just be very careful with that. Also, with this Benedict reagent, you will have goggles, gloves, and you cannot dump it in the sink. Yeah, so you will have a bottle for dumping the Benedict reagent. Few safety tips. When you are using a hot plate, remember to make sure that it is glassware resistant. Remember to make sure that you are not putting it in a really high in the maximum temperature. But this advice mainly is if you're working in a professional lab, when you can have something that is really extremely hot, that is higher than 500 Celsius. In our case, we don't need to make this is not necessary, because in our case you have to put it at maximum, because as is for a teaching purpose. Our hot plate only goes to around 100 degrees, so you have to go to the max. And this is important. You cannot put flammable materials or volatile materials close to the hot plate, so be careful with the alcohol. It has to be relatively far from there. Remember to turn off the hot plate as soon as you finish it. Yeah, so to avoid that. One common mistake that happens is this: that you put the cord, yeah, on the top of the hot plate, or the cable is touching one of these sections. So this cord here is perfect, that is not touching the hot plate. This one is a bad idea, bad idea, bad idea. So be very careful. And one advice that I have to tell you before you start the Benedict test: always label your tubes, because if not, you can make a mistake. As I said before, this is an example of the Benedict test. Here you can see the first one here is a blue one. If the solution remain blue, you will have a negative. This will be your negative control. So when you have water, it should look blue. If you have a reducing sugar in a great amount, it should look red like this. And as I said before, these one are intermediate degrees, so you will have green or orange. If it's positive, it will be red. If it's negative, it will be blue. And one of the common mistakes, or something that you have to be careful, is how to read this table. So here you will have 1, 2, and 3, 4 tubes. So here you will have 4 tubes. here you will need to have your Benedict solution, and this is in milliliters. Yeah, everything is in milliliters. So here you will need to add 3 milliliters here of Benedict solution. So yeah, here you add 3 milliliters of Benedict solution in all of them. Yeah, and how you do it? If you want to do it with a pipette, make sure that you have the pipette that goes from 100 to 1,000 microliters. Yeah, if not, it will take you forever. You can also use a Pasteur pipette that is 1 milliliter. So this is in this case, as the volumes are so big. So here you have to put it 3 times. And so you have 3 milliliters, then you can do it 3 milliliter here. You can do 3 milliliter here, and you can do 3 milliliter in each one of these, and you don't need to replace. You can only use one Pasteur pipette. Yeah, the next step is you add, in the first test tube, for example, here you add only water. So here you will have 1 milliliter, and it's very important that in this case you use a different pipette for this solution. So you will need to use in this one for the blue one, only from this one Pasteur pipette. For this one, you get rid of that Pasteur pipette. Then for the glucose, here for the second tube, 1 milliliter of glucose, and 1 of sucrose here, and of course 1 of maltose here. Good. So remember, make sure, important points, which pipette are you going to use? Remember, and if you're going to, you can use a micro pipette from 100 to 1,000 microliters, or a Pasteur pipette, and use different pipettes for each solution. And then, if it's when you are done with the Pasteur pipette, put it in the weight.